In this project, we were challenged with designing an inexpensive imaging solution to diagnose whether an individual has malaria. The target population selected being sub-Saharan African countries. According to the CDC, malaria is a protozoal blood-based disease transmitted to humans through mosquitoes caused by the Plasmodium parasite family. Infected individuals may present with various non-specific symptoms, including fever, fatigue, and nausea. This makes diagnosis based on symptoms very difficult and oftentimes. With the creation of this new device, diagnosis of malaria is expected to be less expensive, more effective, and more accurate.
Solutions in the past have consisted of RD tests, which are efficient but not accurate, and blood smears, which are accurate but not efficient. However, John Lewandowski, a PhD student at MIT, designed a testing device that can detect malaria in merely 5 seconds using a single drop of blood. He uses a laser and magnet system that reveals crystals in the blood of only patients with malaria. Our design stems from this original idea, and we hope to work in partnership with Lewandowski's company, Boston Disease Diagnostics Group. Similarly, our solution also uses a laser and magnet system. However, it is cheaper, biodegradable, does not require a connection to a computer as it already has a built-in lens, and does not require an external power source as it is solar powered. Our design is both efficient and accurate.
And now I'm going to go through the design of the device and how it works.
The device consists of 7 major parts: a laser, a battery, a solar charger, a biodegradable cuvette, a magnet, a magnifying lens, and a PLA enclosure. To begin, an individual pricks their finger on a sanitized finger pricker. A couple of drops of blood are placed into a biodegradable cuvette. The cuvette is then secured into the center of the device, coming in immediate contact with a magnet. The magnet is responsible for promoting the iron crystals within the blood sample to move in a specific vertical, horizontal, or diagonal pattern, signaling the blood sample contains malaria parasites. A laser is aimed at the cuvette to highlight the pattern of the iron crystals. A qualified medical professional can easily detect the pattern of the iron crystals using the magnifying lens located directly above the cuvette. To further verify the results of the test, sample images of iron crystal patterns are included with the device. Furthermore, a small black PLA box is located behind the cuvette to absorb any stray laser beams. A battery enclosed by a waterproof box powers the laser. A solar charger charges the battery. These 7 parts are kept in a 7x8 3D printed PLA enclosure. All of these parts work together to precisely, accurately, and quickly detect malaria parasites within a blood sample.
Our product consists of some elements of John Lewandowski's design of his malaria testing device. But in order to make our testing device more cost efficient, we added magnifying lenses worth about $12 to account for the motherboard, SD card slot, and LCD screen to be able to identify results then and there, and also as an alternative to imaging, which we believe could be taken through electronic devices that the doctors own. A solar charger and panel were added to account for the countless batteries that the other design would need in order to make it environmentally friendly. Our product's overall cost was reduced by less than half of the original design from $100 to $65.